During this video, I'm going to show how the cell division of the Ostereococcus tauri differs from regular cell division. So what is the Ostereococcus tauri? The Ostereococcus is the smallest free-living eukaryote. It is a unicellular species of green alga. I've made a little diagram to show you what the cell looks like in a microscope. How do cells usually divide? Cells normally divide through the process of mitosis. Mitosis is a 7-step process in which a cell divides to create a new cell. What are the 7 steps? These 7 steps of mitosis are interphase, prophase, enlate prophase, metaphase, anaphase, telophase, cytokinesis. During the first step of mitosis, interphase, the cell is just starting to grow. The black ring represents the plasma membrane. The red ring represents the nuclear membrane. The white things inside the nuclear membrane represent the chromatin, the cell's DNA. The yellow cube-like structures on the left represent the centrioles. During the second stage of mitosis, prophase 1, the chromatins start to form into chromosomes. The chromosomes are represented by the blue and pink structures inside the nuclear membrane. During the third step of mitosis, prophase 2, the DNA was duplicated during interphase, so the chromosomes take an X-like shape with 2 identical sister chromatids. Continuing prophase, the spindles start to form and the nuclear membrane starts to break down. And continuing prophase 2, we get to see the centrioles start to move to opposite sides of the cell, and the nuclear membrane starts to break down even more. During the fourth step of mitosis, metaphase, the centrioles have moved to opposite sides of the cell. The chromosomes have lined up directly in the middle of the cell, attached to the spindle fibers formed from the centrioles. Fifth step of mitosis, anaphase, the spindle fibers pull apart the sister chromatids to either side of the cell. In the sixth step of mitosis, telophase, the plasma membrane comes in to form 2 diploid daughter cells. These cells have 4 chromosomes each, as you can see right here. During the seventh and final stage of mitosis, cytokinesis, the plasma membrane has finally separated and there has been 2 identical daughter cells created. And in this last stage here, these 2 identical daughter cells go on to repeat the same process of mitosis again. So a summary of what I just showed you guys is that every living thing that cell divides undergoes mitosis. That exact same process that I've just showed you is used by every living organism, except for the Ostereococcus tauri. I'm about to demonstrate the division of the tauri and how it differs from every other living thing in cell division. Where's the difference in between regular normal living things undergoing mitosis and the Ostereococcus tauri undergoing mitosis? The difference is during the stage metaphase, when the microtubules attach to the chromosomes. I really want you to look at these little white fibers here. Those are called microtubules. And for the simplicity of the structure, I haven't drawn as many microtubules as there actually would be in this cell, but usually there's twice as many as there are chromosomes. Keep that in mind, that's very important. So if there were 20 chromosomes, there would have to be 40 microtubules. And as you can see here, there are 1, 2, 3, 4, 5, 6, 7, 8 microtubules, and there's 1, 2, 3, 4 chromosomes. So twice as many microtubules as there is chromosomes. How do we know this? Researchers from Caltech have discovered this information. More specifically, a former postdoctoral scholar in Caltech's lab, Lou Gan. He was able to make detailed observations in the tauri. A cell with 20 chromosomes had nowhere near 40 microtubules. Instead, he found about 10 small, incomplete microtubules. When Lou Gan was observing the Ostereococcus, when he noticed the difference, it would have been in the phase of mitosis called metaphase. If this were any regular cell or just any kind of organism undergoing the process of mitosis, there would have been 20 chromosomes and exactly 40 microtubules. This wasn't the case when he was looking at the Ostereococcus. So when Lou had looked inside the Ostereococcus, he was expecting to see 40 microtubules attached, hence there's 20 chromosomes. Instead, he saw 10 incomplete microtubules and 20 chromosomes. So during the metaphase process in the Ostereococcus tauri, basically what happens is chromosomes will bunch together in groups so that they're able to be pulled apart by the microtubules. It's kind of hard to do, but kind of like that. They all bunch together and they end up being pulled apart. Just so the biggest difference is they come together instead of being pulled apart as one. In conclusion, the Ostereococcus tauri changes its division in the metaphase stage. The metaphase stage for the Ostereococcus tauri is greatly different than the metaphase for every other regular living organism. Basically what happens is it has a lot less microtubules than chromosomes, almost cut in half, and the microtubules should be double the chromosomes. So that's the biggest difference. And another big difference is that the chromosomes, in order to be pulled to either side of the cell so that when it splits they have an equal amount of chromosomes, they have to group together and then be pulled by the lesser amount of microtubules.